This video is about Cellular Respiration Lab Part B. You should have already done Part A. The idea behind Part A is that you were using the movement of a red droplet to measure the amount of oxygen consumed by organisms in different scenarios. This, Part B, is very similar. Again, we will be studying the amount of oxygen consumed. However, in this part, we are able to use our Vernier Lab probes, which is a device that hooks to our iPad and electronically collects data. They're super nice. So what you would have in this part of the lab is a bottle that is closed on the top with the probe inserted into it. In the bottle, you would have an organism on the bottom, and we would let it sit there for about 5 minutes. During that time, our probe would be hooked to our iPad, and on our iPad, we could collect data about how much oxygen that organism is consuming during the 5-minute period. Some of you would be able to bring in small pets if you have them, if they fit in the bottle, and it's no harm to the pet. It's always kind of a fun day. You can bring in your little critter, put him in the bottle for a few minutes, and collect data for respiration rate. In this part of the lab, we are comparing a few different types of organisms, so I would like to quickly define some terms for you, which are important when studying respiration rate. Thermoregulation is the idea that different organisms regulate their body temperature differently. If you and I are laying on a beach down south, getting very toasty and soaking in the sun, our internal body temperature really doesn't change. We tend to stay around 37 degrees Celsius or 98.6 degrees. So whether we are in our house, we are laying on a beach somewhere, or even if we're out in the cold, you and I would maintain the same body temperature. You and I would be called an endotherm. An endotherm is an organism that regulates body temp, and that regulation requires ATP. So we are spending energy to keep our body always at the same temp. If I go out in the cold, my muscles start to shiver, which generates heat when ATP gets broken apart. If you and I are laying on the beach, then same idea. We're gonna start sweating, and that will require the use of ATP. So generally, when endotherms are put into hot or cold environments, the amount of ATP expenditure increases. Meaning, whether we are put in hot or cold environments, our respiration rate will increase. You can see that with the mouse. The mouse at room temperature has a respiration rate of 2.37. When the mouse is put into cold, he respires more because he's working hard to keep his body temp warm. When the mouse is put into a hot environment, same idea. He's gonna start sweating, maybe panting a little bit, and that will require ATP. So for an endotherm, whether it gets hotter or colder, the respiration rate will increase. On the contrary, an ectotherm is an animal that cannot regulate its body temp. If any of you have reptiles, snakes, lizards, you might have to have a heat lamp or a heat rack in their cage to keep their body nice and warm and cozy. So an ectotherm would be like a lizard, a fish, insects, organisms that cannot maintain their body temp. Ectotherms are greatly influenced by those enzyme and protein rules that we talked about previously. If an ectotherm is put into a cold environment, it tends to slow down the reactions in the body of that ectotherm, and we can see that when we take a look at the cockroach. The cockroach at room temp has a respiration rate of 0.57. When it is put into the cold, the motion of the molecules in its body starts to slow down, such that the enzymes used in respiration do not work as quickly. So therefore, respiration rate decreases. If I were to put that cockroach in a hotter environment, it is likely that the enzymes and proteins would start to unfold and denature, and I would see again a decreased respiration rate, or maybe that organism would end up dying because the proteins unfold and denature. In the world of AP Bio cellular respiration, thermoregulation is a key idea to understand. Organisms that are endotherms, whether they are put into hot or cold, will increase respiration rate because they require more ATP to maintain their body temp. However, ectotherms will follow the enzyme and protein rules that we talked about before, meaning if an ectotherm is put into cold, respiration rate will decrease because enzymes are not attaching to their substrates as quickly. An ectotherm put into hot will also slow down because the shape of the enzyme starts to unfold.